Tonight, we're very lucky to have Sara Lewis, who is Dr. Sara Lewis, who has a PhD from the University of Arizona. Her mentor, well, one of her mentors, Dr. Michael Kruer from Phoenix Children's and University of Arizona, who is the principal investigator on our genetics study, to provide you with an update on that study and some things that we've learned in the course of that about genetics. So I'm going to take you through a quick introduction of the CP Research Network, and then we'll turn it over to Dr. Lewis to present, and then Dr. Kruer will join us for the Q&A. Okay, so the CP Research Network exists to optimize the lifelong health and wellness of people with cerebral palsy and their families through high quality research, through education, and community programming. So to just summarize a couple milestones since we were founded in 2015, we launched a CP toolkit that's been served up to more than 5,000 people. It's entitled From Diagnosis to Understanding and really helps the community understand what it means to have a CP diagnosis. We later went on to create a well-being guide for parents and caregivers, and then went on to translate that toolkit into Portuguese and Spanish. And then in 2020, we launched MyCP, which is a personalized web portal inside of CPRN's website that allows you to get personalized information, among other things. From a research perspective, we really were founded to create to define a registry for CP to accelerate research. So we defined a clinical registry, i.e. a registry that collects data based on people's visits to a clinic or a hospital setting. That was launched in 2016 and has more than 7,500 patients in it. And then we launched a community registry so we can learn about people's experience from outside of a clinic setting, which we call the community registry. And that was launched in March of 2019 and has over 1,600 participants in it. One of the first things we did was we created a patient-centered research agenda by bringing together about 200 people from the community, culminating in a meeting in Chicago where we arrived at the top 16 research priorities. So that is a community-driven agenda, and that was published and is available on our website. It was published in 2018. We've gone on to have significant public funding milestones for our registry, for this study in genetics that you'll hear about, for work in dystonia and adult well-being. We have also 4 quality improvement initiatives. Quality improvement initiatives are another way to improve outcomes like research is, but it happens to move much more quickly on a different type or sets of problems. So we've got 4 of those going across the network that we launched in 2020. We've had 8 academic publications. So we're trying to, research is about generalizing knowledge, so publishing so people can learn about our findings. Over 27 academic presentations, and we have 5 new manuscripts under development currently. And then we also are focused on wellness, and we launched a virtual wellness program called MyCP Fitness in June of 2021. And we've partnered with the National Center for Health, Physical Activity, and Disability to bring their mentor program, that stands for Mindfulness, Exercise, and Nutrition, to optimize resilience. So it's a great 8-week virtual program aimed at the people with disabilities, but we recruit for the CP community. So what is the network? It's a 501c3, so we're a nonprofit. We organize a collaborative network of hospitals, universities, clinicians, therapists, researchers, and members of the community to conduct research to improve outcomes for people with CP. We have a data coordinating center where these databases or registries are kept, and that is at the University of Pittsburgh's Epidemiology Data Center. We have 2 registries, a clinical registry and a community registry, which I described in our milestones. And then we have our education and well-being programs. So we titled this webinar series the MyCP webinar series because MyCP is really focused at bringing the community together and provides this free portal that offers personalized information and access to other services that we have within CPRN, and anybody can sign up for free there. You can participate in research there. I will say our community registry is currently paused, but it should reopen very shortly. It's been paused while we made our transition to the University of Pittsburgh. There are private discussion forums with clinicians and amongst members of the community about research and lived experience, personalized web sources, resources, and access to our free wellness programs. So just to give you an idea of what these registries are and how powerful they can be, the top two graphs are from our clinical registry back in November 2021 when we had 5,200 patients in the registry. What you see on the left is a graph, a bar chart of gross motor function classification system. This is a very important classification system for people with CP and really describes how they move through the community, either independently, with assisted, with gait assistance, or with a wheelchair. And then on the right, you see the distribution of age bands across the network. So you see more than 20% of the people in the network are actually adults. That's in the clinical registry. The bottom two graphs are the same two data points, but from our community registry back in August of 2022, when we had just under 1,500 members of the community in the registry. So that gross motor function classification system, you see a very different distribution, and that's more reflective of web bias, of what you can find online. And then by the same token, if you look on the right, you see we definitely lean more towards older ages in the community registry, which means we can really answer questions from the community very quickly from the very engaged adults that participate in our community registry. So those are our programs: community engagement in research and quality, education, and health and well-being. We, as a clinical network where our sites are represented by the green pins. There are over 30 centers in the network today. The green pins are sites that are actively collecting data into the clinical registry and enrolling patients in that. The yellow pins are sites that are preparing their site to be able to enroll people. The red is people working through compliance or technology issues. And blue pins are sites that have raised their hand and said, we want to join the network, but they have not been able to get through the process as of yet. So as I said a little earlier, we exist to accelerate revenue, and this is our study pipeline. And what you can see by looking at this is on the left is we have sort of the idea funnel. It's where anybody can bring an idea to the CP Research Network, and we discuss it and we work on moving it along to the point that we approve it as a concept. We submit it as an application to a funder. If we get funded, we go on to implement that study. We conduct that study for some period of time. And then we publish off of the results of those studies. And then what we try to do is take the findings and roll it back in to the care process. So it makes a platform for continuous improvement of the work that we're doing. The blue and green ovals are research projects. The orange ovals are quality improvement projects that we've got. So we've got a lot of work going on throughout the network. So that's an introduction to the CP Research Network. And so what I want to do now is turn it over to Dr. Sara Lewis, who's a postdoctoral research fellow at the University of Arizona in the Kruer Lab. She got her PhD in 2015 in neuroscience at the University of Arizona, and she's going to give you a bit of background about her work and some of our more recent findings. So take it away, Dr. Lewis.
Thank you, Paul. I'm going to bring up slides, and I hope that you got all of the technology problems tonight. No offense.
I hope so too. Thank you.
All right. Can everybody see my slides okay?
Yes, great.
Wonderful. Thank you. So I want to say thank you to everybody tuning in, and thank you to the CPRN for having me. As Paul mentioned, I'll be talking about cerebral palsy genetics, and in particular, I want to emphasize some of the opportunities this has created, both for research as well as clinical applications. So a little bit about me. As Paul mentioned, I am a research scientist. I have a PhD in neuroscience, and my focus is on using gene findings from individuals with neurodevelopmental disorders to understand how the brain is supposed to develop and what is going wrong in those particular disorders. And as part of that research, I use a combination of patient-based studies as well as Drosophila, which are these fruit flies shown here, kind of walking up in one of the assays I use in the lab. And another thing I wanted to note is that I'm the mother of a child with a hypermobility disorder. So making sure that the research is approachable to the patient population and having a clinical impact is a big motivator of mine. So to kind of talk you through the concepts that I'll be touching on, I'll be addressing some key questions. So what are cerebral palsy genetics? How are they studied, and what are some key findings? What can genetics tell us about the changes to the brain in CP? And what is the benefit for identifying genetic causes for an individual with CP? And let's get this out of the way right now. If anybody's freaking out about genetics, thinking about the plants and the squares, we're not talking about that at all. So what I'm going to be focusing on is the changes to the gene. So we're interested in variants. Variants are misspellings in the DNA that create abnormal proteins. So our lab is interested in, say, detecting variants and then asking the question, well, what is the consequences on protein function? And what are the consequences in cellular function, brain function, developmental processes, all of these sorts of things. And the other thing that we really key in on are what are called de novo mutations. So de novo mutations are new. They arise in the germline, which are the sperm or the eggs, and are transmitted to the affected individual. So you don't need to keep track of squares or wrinkly peas, none of that. So why do we think genetics are important in cerebral palsy? Because for years and years, this was not something that was thought about. And there's a couple of key things that came out to make us think that this might be an important thing worth looking into. Number one, about 30% of people have no risk factor or adverse event in their history that could explain why they acquired cerebral palsy. It was also noted that there was an increased risk with increased paternal age, and that goes along with de novo mutations. Another observation was that the prevalence was 5 times more common in twins than in singletons. And when you look at registry data from an epidemiology standpoint, the heritability or the ability to get genetic inheritance was estimated to be about 40%. And so with time, some more studies came out looking at families that had multiple affected individuals, and Mendelian gene inheritance patterns were described. And ultimately, this led to some more advanced genetic studies, such as whole exome sequencing and copy number variant studies. And, those are done in cohorts, large groups of patients. And that's going to be where I really focus today, talking about whole exome sequencing studies. So what does this look like? So first off, the exome are made up of the exons. So that's the part of the gene that actually gets spliced into the mRNA and then translated to form the amino acids that make up the protein. So the exome is about 30 million nucleotides, which is 1% of the genome. So it's a lot to keep track of, but it's still only a fraction of what is there. And in whole exome sequencing, the DNA is copied many, many times, and then it's sorted to match the known reference genome. And you can look at this, single sequence, and you can ask if it's different from what is observed in the population. And you can also compare an affected individual with both parents and see if there's a de novo or a new mutation as well. And so if you're to look at the peer-reviewed papers that have been put out, we're seeing that the studies for cerebral palsy genetics is increasing every year. And there's now a number of whole exome sequencing cerebral palsy cohorts that have been described and published. And this makes it possible to do what's called a meta-analysis. So this is saying you have multiple observations, and so you can say something about the population as a whole using some modeling techniques and math. And so having the ability to do a meta-analysis allows you to ask, some of these big questions. And one of the biggest questions that is on everybody's mind is, what fraction of cerebral palsy is actually due to a genetic cause? And so this is diagram showing the output of that study. And so this kite shape is describing the range of possible results. And that wide middle bit is showing the average amount. So the percent identified genetic cause going left to right there. And so when you look at this group as a whole, it's around 23%. And this is a pretty conservative estimate, actually. And this is taken from a large set of data, 11 articles and about 2,500 patients. And, the other thing that was noted looking at this population is that if your combined is about 23%, if you're talking about people with no risk factor, it's going to be higher, around 35%. And if you're talking about individuals with risk factors, it's lower, around 7%, but still not zero. Another big question that was addressed in this meta-analysis is, well, what are the genes? What genes are actually causing cerebral palsy? And this yielded 258 genes. So this is specifically describing genes that had what are called pathogenic variants. And a pathogenic variant is a change that is thought to be the actual cause. And so what was noted is that, you have a large list of genes, very few of them are seen in more than one patient. And so they're individually rare, but collectively as a group, common. And in addition to the genes that were actually described here, this 258 genes, if you go to the original publications, you'll note that there's hundreds of candidate genes that haven't been definitively, pinpointed as pathogenic. And so we're only scratching the surface of what these genes might be. And as a researcher looking at a list of genes like this, what comes out at me is that these are very heterogeneous. These are very different. And so this really raises the question of, well, what is it that these genes have in common? Like, what are they doing and why is this happening? And so to look more at that, we turn to a branch of science called bioinformatics. So bioinformatics is that intersection between statistics and biology. And the idea is to look at large sets of data and try to figure out what those common elements are. So this includes things like, shared molecular functions, interacting in the same cellular compartment, being expressed in the brain at the same time and place, or links to known disease states. And so looking at these lists of candidate genes, we were able to ask some questions. And one of the big ones that we started with was, are cerebral palsy genes related to the genes that cause other neurodevelopmental disorders? And those are autism, epilepsy, and intellectual disability. And the reason we had this hypothesis were two major reasons. One is that neurodevelopmental disorders are known to co-occur with cerebral palsy. And the other is this idea of overlapping brain region involvement. And so what do I mean by that? So in each of these different disorders, there's different domains of development that are affected. Cognitive development, language development, social-emotional, and physical development, or your movements, right? And in a typical brain diagram, you'll see something like this. So you'll see, oh, well, here's the motor cortex, shown in orange, and here's the frontal cortex shown in green, and here's the part of the parietal lobe in blue that we know regulates language. And this seems simple at a first pass, but when you look inside the brain, it becomes immediately apparent that there's a lot more going on. So it's not one region of the brain making movements happen, because it's connected to all of these other parts to process the information and to take in information. And this information is important because this is your sensory information. This is your ability to adapt to a changing environment. This is your ability to modulate your movements and say yes or no, depending on the situation. And, when you think about movements, it's not this little slice of the brain, but it's distributed throughout. And if you start mapping on some of those other domains of development, you're going to see that they're overlapping, that they're right next to each other, and those connections are, very interconnected. And so what bioinformatics was able to tell us is that these CP candidate genes are strongly linked to other neurodevelopmental disorders. In other words, it's all interconnected. And so if you were to look at these gene lists, instead of falling over here as cerebral palsy genes, they're going to be falling into these other regions as sort of a broader context of neurodevelopmental disorder genes. So we asked what else these genes have in common, and it turns out that it's making connections. So we found that a lot of these genes had a known molecular function in axon extension. So the axon is the output of the neuron, and it's shown up here on the top left. And the idea is during development, a really important step is extending those axons and making the right connections. And those connections are going to be what drives that ability to have the information at the right time and place to process the information appropriately, as conceptualized in this video here. And again, I just want to sort of talk a little bit about, and when you have candidate genes, that opens up a lot of possibilities for research. So as I mentioned, my research also uses Drosophila. And when we look at one of these candidate genes called AGAP1 a little closer, we did indeed find that those axon terminals were much smaller than they should be. And so that's decreased connectivity between the motor neuron and the muscle. And, we could look even closer and find out that there was problems with the way proteins were being moved around in the cell, and that increased protein turnover, and that increased sensitivity to cellular stressors. And so looking at this gene function in the models really gives more insights into what's going on at a basic level. And there's studies like this going on all over the world using all sorts of different organisms. And so kind of thinking about big picture from research, what is it that these genes are telling us about the brain? It's really pointing to this connectivity idea. So this is looking at a map of the typical brain, and the connections that go in different directions are shown in different colors. So left to right is going to be green, front to back, or sorry, red, front to back is green, and then top to bottom is blue. And so what we're finding is, very similar to other neurodevelopmental disorders, there's something about disrupted connectivity during brain development. So we're going to switch gears here and think instead about clinical applications of genetic findings. And one of the points of interest is what's called precision medicine. So precision medicine is thinking about using information about how a particular individual is affected and using that to guide clinical decision-making. So if we think about a population, there's going to be some people who respond very well to conventional therapies and others who don't, and nobody's going to look the same. And if you had a way to identify who these subsets are, who's going to respond well to replacing or targeting a specific gene function, or who needs, constant surveillance or preventative strategies as part of their care, or who's going to be a good responder for a known effective treatment that works in a subset of the population. So this is the idea of precision medicine, of finding the people who need that specific type of care that is outside of the typical treatments. But to think about it more concretely, I'm going to tell a success story. So ADCY5 is a gene that has variants that increase the activity of that particular protein. And when those variants are present, you get unwanted movements, also called dyskinesias. And this ADCY5 protein is activated by this receptor, and this receptor is activated by a neurochemical called adenosine. And you may not have any clue what adenosine is, but you know this receptor because this is where caffeine goes. And when caffeine is present, it blocks the activity of this receptor. And so the idea is, if you know that there's too much activity in this protein here, and you know that it's reliant on this receptor, maybe you could block this unwanted movement by blocking this receptor using a known and safe antagonist. And sure enough, doctors tried it and had a great deal of success in individuals who have this particular genetic disorder using coffee. So you wouldn't normally give a child a cup of coffee to help them go to sleep, but in this case, it's the right call. And you wouldn't know that if you didn't have that piece of genetic information. And so thinking about it from a research standpoint, we're asking for individuals who are receiving clinical sequencing as part of their care, who's going to have a potential personalized finding, personalized medicine finding. And so we are calling these clinically actionable findings, and those include, targeting that primary disorder, prevention strategies, or specific symptom management. And when we looked at this set of published variants, what we found is that 7.1% of individuals who received clinical or research sequencing had a finding that would change their care, potentially improving their outcomes. And of note, another 19.6% of patients had some other genetic finding. So this was presented at the American Academy of Cerebral Palsy and Developmental Conference, and we're anticipating to publish it shortly. So I mentioned a couple of key concepts, and that's the idea of a potential genetic cause, studying it from a both big picture aspect as well as the specific gene function, and then clinical interventions. But how do these tie together, and what does this look like going forward? And so, as I mentioned, there's going to be some proportion, we think somewhere between 23–40%, have a genetic contribution to their cerebral palsy. And in some proportion, you actually identify that genetic cause. And once you have that, you can learn more about the gene pathway function from a bioinformatics standpoint. You can take it to your fly in your lab and do functional studies. And from there, you can understand what is actually changed and what potential options exist for modifying function back to typical function. And from there, you develop your clinical interventions, you trial it in patients, and you may potentially have, improved outcomes in the clinic. And so that's the translational pipeline. But as I mentioned, it's leaky. We're still not detecting all of the genetic causes, and we're still trying to understand what these genes do so that we can develop these interventions. So what is the future of genetics? So shown here in this picture is a conceptualization of CRISPR. So CRISPR is, in the short term, DNA scissors, where you can cut out a piece of DNA and swap it out with something else. And this is really revolutionizing what we think is possible for genetics. And I don't know when it's coming or what it's going to look like, but it is very exciting. But I can also say closer to home, there's already, changes in certain neuromuscular disorders that are hitting the clinic that are using genetic approaches to try to help patients. And those include spinal muscular atrophy and Duchenne muscular dystrophy. So the hope is that some genetic causes of cerebral palsy might have treatments like this in the future. And so the question of, are our genes our fate? If we have this genetic finding, is that the end of the road? And I would instead say no. So the relationship between genotypes and the expression of the traits, or what the outcome is, isn't always straightforward. And there's just a lot of steps that happen along the way. And one of the things that we know is that early interventions, physical therapy, still remain some of the most important tools that you can have. And so I would say, one way to think about this is the first step in a different type of journey for both researchers as well as patients. It might create new opportunities with, the right information. So in summary, as I mentioned, we think about 23% of cerebral palsy is likely genetic, although that number could be much higher. These genes have lots of different functions, but they seem to be regulating brain development. And one of the themes that is emerging that I touched on is that connectivity matters. The other thing I mentioned is that genetics can help understand cerebral palsy better and develop new therapies from laboratory research partnerships. And that genetic sequencing can be really important for helping understand why no two children with CP are alike, but it also allows you to find groups who might benefit from personalized medicine. And so I just want to say really quickly, thank you to everybody who makes this happen. And that includes the patients, the collaborators, and everybody who supports these studies. So I'm going to turn it back over to Paul for some Q&A.
Great. Thank you so much, Dr. Lewis. That was, I have to really hand it to you. I do have some things that I want to ask you to sort of spell out, but that was really clear. And as much as I've watched Dr. Kruer present, I learned even more, and I really want your puzzle slide because that's really helpful for the importance of explaining precision medicine.
Yes.
So I think you said mRNA without saying what it was, and I was wondering if you could just say what mRNA is and how it relates to the exome and the gene and just to tease apart because it's an important term.
Absolutely. So when you have your original genomic DNA that lives in the nucleus, your first step is actually transcribing the gene, which is making a copy of it that's called RNA. And then that RNA is sort of like packaged up for, easy shuttling around and working with. And that's called messenger RNA. So that's the mRNA. And that's sort of the ready to be translated into proteins in the cytoplasm of the cell, so outside of the nucleus.
Okay, great. Thank you. I'm sure there are others, but I'm certainly not going to dominate. So let me first of all invite Dr. Michael Kruer. Let me see if I can actually, I haven't done pinning in a while, so let me see if I can pin the two of you so you can be available for Q&A. But I want to open up to questions. You can either enter a question into the chat, or you can unmute and ask a question, or you can raise your hand and I'll call on you. Any of the above, I'll do my best to manage not having pandemonium. Let me pin. Oh, I can only pin one person. Well, that means Dr. Kruer gets to speak if the question is for him, and you are on point, Dr. Lewis. So, okay, questions. I won't let this go too long without asking more questions. So I'm gonna keep it going. People should raise their hand or type a question. So I made a very brief explanation in the blog post about why sort of fly models are important. Can you say a little bit about like whether or not there have been examples of studying genes in flies that have been translated to humans, or how does that translation work?
Yeah, so one of the top ones that come to my mind is epilepsy. And so when you express epilepsy causing variants in fruit flies, and there's very good gene conservation, so they, work in the brain the same way. The flies do have seizure-like activity, and that was used to look at a bunch of different pharmaceuticals. And I don't remember exactly which ones have been demonstrated to be successful, but some of the most popular NMDA receptor antagonists, I believe, were initially trialed in fruit flies and then moved into patient studies.
I'm just smiling when you say MDNA antagonist. So, yeah, so great. So so flies have been used as a model by which you can translate something that happens into a fly into an actual like medication pharmaceutical for humans safely.
Absolutely.
Yeah, I think that's a really important part for people to understand. Okay, we have a hand up and a question. I know I know Susan Picerno has a burning question, so I'm going to let her go first, and then I'll go to the chat. You're muted, Susan.
Thank you. I appreciate the opportunity to talk. I both greatly appreciate your research project and am terrified by it. I think about it all the time. And of course, right at this moment, there's someone at my door and my dog is barking because he can hear the person at the door. Can I put myself on hold and then
why don't you
come back?
On hold, we'll come back.
Thank you.
Yeah. I mean, Oh, I'm not being able to mute you. That's not good.
Let me try to mute me.
Yeah, since I don't seem to have the usual controls I should have. Let me reclaim host and then I can mute people. Okay, so Michelle asked a question. How do we advocate for CP genes to be included in genetic testing?
So Michael might speak to this a little bit more, but there's no, linear answer for how a gene ends up on a panel. But I think the word that's being used here, advocate, is absolutely a big part of that. So part of it is, recognizing that this is an important contributor to cerebral palsy, that, people want to be able to detect it. So that's also part of why the precision medicine piece we feel is very important. Making the case that this is not just a curiosity, but something that actually has clinical consequences, in the positive way of consequences. So I would, work with advocacy groups and see what's available for, bringing this to attention and, having insurance companies pay for it and clinical sequencing companies put it on their panels. But I do know that there is this whole process and they are receptive to advocacy.
Okay, great, thank you.
Susan,
I think Michael wants to
go
say
ahead.
something on that.
Briefly add to that, Paul.
Yeah, sure.
So I certainly agree with what Sarah was saying. But one of the things that physicians will typically use when deciding whether the patient right in front of them gets something like genetic testing are clinical guidelines. And these are often in the form of what are called practice parameters. They're based upon a great deal of research and evidence, but there's something that expert committees will comb through that evidence and then make an official recommendation. And so it's still quite common that physicians are diagnosing patients with cerebral palsy every day and trying to determine what testing is appropriate for that child or sometimes that adult. In the past, the practice parameters have said something to the effect of there is no role for genetic testing in the workup of individuals with cerebral palsy. We now know that that's not the case, that's out of date. And so there are current efforts that several members of the CPRN are involved in, including our group, that are in process to try to update those recommendations. And I think that that's one of the best ways to bring genetic testing to patients and families with cerebral palsy is just to update what those expert recommendations are for practicing physicians.
Great, thank you, Dr. Kruer. Susan, go ahead, give it a shot.
Thank you very much for the second chance here. I very much appreciate what you're doing and honor your interest in the health and well-being of my community. I also understand that this is the research of the future and that basically we have to understand the genome to make progress. That being said, I'm terrified by the practical outcomes of the research in the sense that in the past, to give one example, folks with Down syndrome, when they were able to recognize which children in the womb had Down syndrome, now 90% of those pregnancies aren't brought to term. And that also has impacts on the ultimate research, because there was an article that I posted on CPRN of a gentleman who was Indian American, and he decided to go forward with a pregnancy of a daughter with Down syndrome. And then he was a genetic researcher, so he decided to do research into Down syndrome. He actually found, I don't exactly know what the formulation was, but something that made their brain connections work better and ultimately made them a little bit smarter or somewhat smarter, because this medicine, let's call it, made their brains work a bit better. So he went to a pill developing place. I'm sorry, I'm blocking on the word at the moment. Thank you, a pharmaceutical agency. And they said to him, well, why would we ever develop this when the people you want to develop it for, quite frankly, no longer exist? And it took him, according to the article, it took him 10 years to find a pharmaceutical company that was willing to manufacture this, even though there wasn't side effects or maybe any negative outcomes. They just couldn't see the market for it. And ultimately, he was able to manufacture it by using rare disorder funding. But in a sense, that was a shame, because that is supposed to be used for people who have actual rare disorders. And so that was somewhat of a misuse of the funding. So I don't know how we get across to the general public that it would be helpful for them to hold on. First, we find the genes, then we need to have a little bit of lag time to make the positive outcomes happen. And they will happen. And I believe that you will make them happen. But because there's that lag time between the time when we find the genes and the time when the outcomes happen, it can have some really negative outcomes for the community, and I'm concerned. And thank you for listening.
Any comment that you want to add to that, Michael or Sarah?
Yeah, well, I really want to thank you for speaking to this important issue, and the ethical considerations and are absolutely something that is being discussed heavily in the research and clinical community. Like, it is absolutely something on people's minds and that they're trying to get ahead of. But also that point to, it's a process, it's a pipeline, it takes time, is something I try to emphasize in my own communications. And thank you for recognizing that, because again, that is another key issue of it takes time. So let's get started. So thank you.
Thank you. Amber, you're muted.
Sorry.
That's
There's
okay.
so many different causes of cerebral palsy, trauma being a major cause, infections, etc. I'm just wondering, how long has it been speculated that there's a genetic component? Because a doctor that I had said that there's always been like a 5% genetic component to CP, which I wasn't aware of until a few years ago. So is it just now being more recognized than it was before, or I just want to understand that part of it, and the link between other developmental disabilities with that.
I know Michael has like a razor sharp understanding of the history, and so I'd love him to tackle that part of the question for sure.
Yes, so I think that our understanding of the causes of CP is definitely in flux. As you mentioned, Amber, we've known that birth-related traumas, difficult starts to life, are very common for individuals with CP. And I think that there was this concept that genetics could account for a very small proportion of CP cases up until probably the last 7 to 10 years. And I would say within that time, we've just, as Sara was showing with her slide, we've seen this explosion of interest in the community, in the research community, and a growing recognition that sometimes genetic causes, genetic mutations are the cause of cerebral palsy. What I think is still very much up in the air, but is a fascinating and important question is, why do some kids have a difficult start to life? When do genetics and environment interact and how can understanding these different aspects help us to care better for people with CP throughout the lifespan. And I think that that's where the research is really headed.
Have you seen within families, like say if a second cousin has CP and then, then another family member, is that more common if there's like a cousin removed or something like that has it?
So I think that the most common pattern that we see, and Sarah spoke a little bit to this, was that there's no one else in the family that has cerebral palsy. And I think that's what threw us off for so long, until we realized that these spontaneously occurring, out of the blue de novo mutations are actually one of the most important modes of inheritance for CP. So mother doesn't have it, father doesn't have it, but yet the child has a genetic change that in fact leads to their CP. That's the case for, I would say, most families in America, but I actually personally got interested in the genetics of CP back when I was a medical trainee, and I met a family who had 4 children who all had CP, and I had to stop and go, that's kind of unusual. And that's what first got me thinking about this, and first led to the light bulb that genetics might actually be relevant for cerebral palsy.
Oh, that's very interesting, because I knew a family who, like, a second cousin had it. So that's why I asked that. And I thought, just to piggyback that, how is the genetic component going to affect like kind of like having a pre-existing condition? Will that be a negative effect, do you think, in the long run for insurance stuff and getting things covered for people with cerebral palsy?
Well, one of the things that we and many others have been emphasizing with this work is that we're not undiagnosing people with cerebral palsy, that cerebral palsy is a recognized condition. It's a true clinical diagnosis. And so what the genetic piece is doing is it's giving us as researchers and as doctors, and it's giving patients and families more information than ever before to help close the loop and understand the whys. Now, like Sarah was saying, one of the exciting recent realizations has been, gosh, even right now, that actually changes how we treat patients or recommendations, for roughly 1 in 4 patients with a genetic cause for their CP. The question of whether that can impact insurance and pre-existing conditions, it's a complicated question. It's definitely something to discuss with your own healthcare provider if that's on your mind. But there are laws. There's a law called the GINA law, Genetic Information Non-Discrimination Act, here in the United States, that protects people from discrimination in their healthcare and their insurance related to genetic information. There are some limitations to that, so definitely discuss that on an individual basis.
Okay, thank you. I was just wondering.
So before we wrap up, since I had a little bit of technical difficulty getting us going, there is a fairly technical question that if you guys could answer, which is how and why are VUCS found during genomic analysis reclassified as pathogenic? And if one of you could answer that, I would feel like we're done.
Just nail it. Yeah, so I would be happy to take it. So a VUS is a variant of unknown significance. And I think that right there in the name really explains why it might eventually be classified as either benign or pathogenic, because that label means we don't know. We found this change. We can tell that it's maybe not frequently seen in the population. Maybe it wasn't seen in a parent, but we all have de novo mutations. Which ones are actually important? And so as research happens, as more clinical information is being shared, it becomes easier to interpret those. So what we do as part of our work is, we talk to clinicians around the world who have patients with disorders in the same rare genes and ask what they're seeing in their patient. We do studies to understand how that variant is changing the protein function. And from there, we might be able to say, oh, this one actually does have a change in what the protein is able to do. And so, with more information from both the research as well as the, sharing clinical data, not clinical data, but, having that conversation about the fact that it's being seen more than once allows us to then reclassify variants like that.
Great. Well, I want to thank you, Dr. Lewis, so much for taking a very complex topic and making it very consumable. Dr. Kruer also for both supporting Dr. Lewis as well as being available for Q&A, and everybody for attending. So thank you, and nice job kicking off the MyCP webinar series for 2023. Thank you very much.
My pleasure. Thank you.
Bye all.